Okay. Well, okay. Let's begin today's session. Hi, thank you for joining. So today we will be discussing about glycolysis, partly about gluconeogenesis, and then we'll touch upon TCA, tricarboxylic acid cycle or citric acid cycle, right? So today's topics are not like we can discuss in length because these are pathways, right? So what you need to do is you need to remember the pathways. You need to remember which substrate get converted to what form, and then how it is linked to the other next step of the pathway. So the pathway in itself will take one class each if we are to go in details of each of these pathway. So we won't be doing that because this is a problem solving session, and more than this it's a revision session. So what I will be doing is, of course, there I have all the pathways, like all the steps in the slide. So what I will do is I will just mention the step. I will tell what major form is converting to what minor major form, which enzyme is being used, and if there is something particularly important about the step. But do refer to some book so that you understand each of these steps in a much detailed way, right? But having said that, there are many questions in today's session which will help you get a grasp over these topics as you go ahead. So my only cue to you is today's session will be a bit different. It'll be more about like looking into the slides and reading and like looking into the text. So we won't have many numericals or questions to solve for that matter, but that's just about it, because that's these topics are meant for you to like remember it in the terms of the reactants and the products. So that's what's going to be about today, but we'll try to make it interactive. We have some questions, and I have put the questions in between so that when one topic gets over, we have the relevant questions after that, right? So that makes it more understanding easier. Okay, so like you can see on the slide, it's mostly about glycolysis in today's session. So let's begin. Glycolysis is basically, in very simple terms, as you will also understand, glycolysis is basically breakdown of glucose. So you are taking in food, and now you want to use that glucose up. You basically say that I need energy, and that's why you are eating food, and you are taking in lots of glucose, or in other forms of sucrose that's broken down into glucose. Now glucose is what's going to give you energy. So we are going to look into how glucose gives rise to, how glucose is um, broken down so that you can get to the step of energy being ATP generation, right? So we'll not go to ATP step today, but we will cover how glucose breaks down happen and what happens immediately after that. Okay, right. So the breakdown of glucose is called glycolysis, and glycolysis has 10 steps. The 10 steps are divided into two phases. First phase is called preparatory phase. The second phase is called payoff phase. We will see each of them in the slides which we will discuss today. So preparatory phase is basically making the glucose amenable to the conversion for ATP. And how does that happen? So in preparatory phase, what actually happens is, just give me a second. Yeah. So in preparatory phase, what happens is your glucose is phosphorylated and it is converted to glyceraldehyde 3-phosphate. So this glucose to glyceraldehyde 3-phosphate, this is not one single step. There are chain of reactions that takes place in these conversion, and those steps are what makes it ready, or the glyceraldehyde 3-phosphate is what is to be used for ATP generation. So the phase is called preparative because of preparing your glucose for ATP generation. And you have these 5 steps. Each 5 steps here are marked by 5 enzymes, right? So you have glucose. So let's begin. The first step is glucose. Glucose uptake, you would have heard glucose is taken in, glucose is taken into the cell. So we have the glucose in the cell right now. What happens? The very first thing that happens is glucose is phosphorylated. Glucose is phosphorylated to glucose 6-phosphate using which enzyme? hexokinase. Now, hexokinase will add a phosphoryl group here using ATP as its energy currency or ATP as the driving factor. Okay. Now, in this step, what is this called? This is called activation of glucose. So, first step in glycolysis is glucose activation. Okay. And this is an irreversible step. Okay. What is happening in this step is basically it is transferring the terminal phosphoryl group from ATP to glucose. So one phosphoryl group from ATP is being transferred to glucose and it becomes glucose 6-phosphate. Where is the phosphoryl group getting attached? It is getting attached at this sixth carbon end. Okay. So at this end, you have your phosphoryl group attached. That's the first step. That is also called priming reaction. It is also called activation of glucose. So that's the very first step that you need to remember. Next, once you have the glucose 6-phosphate ready, what happens after this is it isomerizes to form fructose 6-phosphate. This is that step. This step again uses ATP. Okay, but this is reversible. So here an aldose, which is your glucose 6-phosphate, gets converted to a fructose 6-phosphate, which is a ketose. So there is an aldose to ketose uh, isomerization. That's because of the rearrangement of carbonyl and hydroxyl group at C1 and C2. So your C1 and C2, these carbonyl and hydroxyl group get rearranged. So your six carbon ring membered ring goes to a structure which has a five edges. Okay, so you have a fructose 6-phosphate from a glucose 6-phosphate. This is isomerization reaction. Okay, and this is again reversible. The first step is irreversible. Just a second, sorry. So the first step is irreversible, that you need to remember. These steps are reversible, right? Now, how did this rearrangement take place? How did a glucose 6-phosphate, which looks like this in structure, get converted to fructose 6-phosphate, which looks like this in structure? So here you can see the reactions. So this is how your glucose 6-phosphate is. The first thing that happens is opening. If you remember, we have looked into how closed ring structure can form open ring structure. So the closed ring structure has formed an open ring structure. Key for you, remember what does opening and closing relate to, right? In which concept did we learn about opening and closing of rings? Cue for you to remember that concept or topic that we discussed. Okay. Meanwhile, while you're discussing doing that, so you have your ring opening. So the first thing that happens is this ring opening. So what is happening here is you have your CH2, which this is your first here, and then this is your sixth. Okay. Now in the second step, you have an attack by the enzyme. Here you have the H plus being donated. This H plus forms something called enediol intermediate. What is en? En is this. What is ol? This is OH. So enediol intermediate. And this leads again to a rearrangement, which is forming the final structure of fructose, and then this gives rise to fructose 6-phosphate. So there is an enediol intermediate between glucose 6-phosphate and fructose 6-phosphate. That is what the last line here referred to, rearrangement of the carbonyl and the hydroxyl groups, right? So you have fructose 6-phosphate from your second step. Now what happens in the third step? In the third step, your fructose 6-phosphate gets converted to fructose 1,6-bisphosphate. What is 1,6-bisphosphate? It means there is another addition of phosphate. You had already a phosphate added onto glucose in the very first step. Again, in the third step, which is here, you have the second ATP that is being used. By mistake, I said in the previous slide that there is an ATP used in the second step. That's not the case. That's an isomerization. In the third step, you have ATP, which is being used for priming the fructose 6-phosphate. So wherever you have ATP being added to the substrate, it is called this priming reaction. So you have an ATP here that is being added to the fructose 6-phosphate. That leads to the formation of fructose 1,6 bisphosphate. Which enzyme? Phosphofructokinase 1. Remember this enzyme. This is PFK1. PFK2 is also there. So don't get confused. PFK1 is what helps or what catalyzes the conversion of fructose 6 phosphate to fructose 1,6 bisphosphate. Now, what is important about this step? This is again an irreversible reaction, and this is the first committed step in the glycolytic pathway. Mean when this reaction takes place, the pathway has committed itself to glycolysis. This product here, fructose 1,6 bisphosphate, ensures that it is the further steps will be followed for glycolysis. Up till here, you can have rearrangement or it can go into some other pathway. But here, once it becomes fructose 1,6 bisphosphate, it is committed to being a glycolytic pathway, right? And it's the first of those. Okay. So you have third step, which is fructose 1,6 bisphosphate generation. Now, what is fourth step? Your fourth, which is this here. It's basically your fructose 1,6 bisphosphate. It's basically cleaving. So the structure is broken down into two further compounds, which are dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. This is because of the enzyme aldolase. This reaction, which is the cleavage of fructose 1,6 bisphosphate, is again reversible. So you can see the here that your enzyme aldolase is catalyzing this reversible reaction. Your fructose 1,6 bisphosphate is cleaved into dihydroxyacetone phosphate and glyceraldehyde 3 phosphate, right? So this one is again reversible. Now, what is happening here is suddenly you have two new products coming up. It's all this while one substrate, one product, one substrate, one product. At fourth step, you have two products, which are glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Now, what happens in the fifth one? In the fifth one, these two phosphate cannot, they don't stay individually. So what happens is there is interconversion of these triose phosphate. So the dihydroxyacetone phosphate converts to glyceraldehyde 3 phosphate. You can see here. Okay, and what lets it to happen? Triose phosphate isomerase. What does it do? So why is it? Because only one of the two triose phosphate formed by this enzyme can be directly degraded in the steps of glycolysis. And which one is that? Glyceraldehyde 3 phosphate. So what happens to the dihydroxyacetone phosphate? It basically gets converted to glyceraldehyde 3 phosphate by triose phosphate isomerase. Okay, and this happens again reversibly, yes, because they are interchanging in forms. But what you need to remember is you started with one glucose and you ended up with two glyceraldehyde 3 phosphate. This is very important. Remember the number of certain uh, compounds, glucose. So if you have one glucose, in the first phase, which is preparatory phase, you end with two glyceraldehyde 3 phosphate. To where the second glyceraldehyde 3 phosphate is from a dihydroxyacetone. Okay, that gets isomerized and it forms the glyceraldehyde 3 phosphate. So that forms the first phase, which is the preparatory phase, right? Okay. So since we have finished the first phase, let's try to look into some questions and try to answer this. Okay. So this question says, which of the following is not a major fate of glucose in the cell? So whenever there is glucose uptake, which of the following pathway is not a mandatory thing to happen? Is it oxidation to pyruvate via glycolysis, so basically glycolysis, or via pentose phosphate pathway, so this pathway, or storage in the form of glycogen storage, or glycosylation of proteins? So which of these is not mandatory, or which of this is not compulsory, or which of this is not as the question says, major fate of glucose in the cell? C, glycosylation of protein. And why is that? As Aditi guessed it right. So the first one says Oxidation to pyruvate via glycolysis is exactly what we are studying, right? So your glucose breaks down to form pyruvate via glycolysis pathway, and it does take place. So that's a major phase. The second pathway is pentose phosphate pathway, via which it gets oxidized to ribose 5 phosphate. This is used for nucleotide synthesis. So that's also a major phase. Fourth one here is storage in the form of glycogen, which is the opposite. If you are not using glucose, you are storing it as a glycogen for further use. So that's also a major fate. Either it is being used up or it is being stored. But glycosylation, as we have already looked into glycosylation, is basically when a sugar molecule is added to a protein. And that sugar adding is not a major fate of glucose. So glucose derived sugars are definitely used in glycosylation, but when you intake glucose, that is not a major fate. That's one of the, how do I say, that's one of the use of glucose, when one of the ways that glucose is used, but not one of the major fate of glucose in the cell. Well, that was correct. Next question. Which of the following statements are true? The first step of glycolysis is the activation of glucose by phosphorylation. The first step of glycolysis is catalyzed by the enzyme glucose phosphatase. In the first step, glucose is converted to glucose 1 phosphate. And in the cell, ATP and ADP usually exist in complex with magnesium ions. So we just discussed it. So this is again very simple. But questions like this is what will make you more thorough with these kind of pathways. Okay. A and D, like Aditi mentioned again. So uh, as you can see, the first step is so the first point is the first step of glycolysis is the activation of glucose by phosphorylation. That is exactly what we did. So that we know is correct, because we remember glucose is phosphorylated and it becomes glucose 6 phosphate. And because the product here is glucose 6 phosphate, that makes the third statement wrong. The third statement said it is converted to glucose 1 phosphate, and we know that the phosphate is added to the 6 carbon, not the first carbon. So glucose 6 phosphate is the correct answer. While the enzyme that does that is not glucose phosphatase, it is hexokinase, right? And in the cell, ADP and ATP usually exist in magnesium ion. That's also correct, because I don't know if it's here. Let me just see. Right here. So here you will see in the first step, when you see this reaction, you will see an Mg2 plus with ATP. So it's basically trying to say that ATP, and we also did it in the previous class, that ADP and ATP in cell is usually found to be complexed with magnesium. So that's the correct answer. Okay, the third one. So out of these 6 statements, which of these is false? Read the names of the compounds properly in the options and then select your answers. E. Is that the only answer? That's it? Do you want 1 minute more so that you can look into the options once again? Okay, cool. So it says which of the statements are false. So E, you got right. Let me just go over the others quickly. So The first one says the second step of glycolysis involves irreversible isomerization. Let me go back to second step once again. Reversible, right? So that's why the first statement is incorrect. It is not irreversible. Which is irreversible? The first step is irreversible, and the third step is irreversible. Isomerization is not irreversible. So this is a wrong statement. Second statement is correct, as we have already seen. How does the carbonyl group transfer from C1 to C2, and that leads to the formation of glucose 6 phosphate to fructose 6 phosphate. That's correct. Third, again, we have looked into this how an enediol intermediate is formed. Fourth, the mechanism employed by phosphohexose isomerase involves phosphoryl group transfer. If it is an isomerase, it does not involve a group transfer. There is no phosphor transfer here. There is no phosphor. Where did we see the isomerase? We saw it here. Here. And here there was no phosphoryl group involved. So that's why this is incorrect. Right? Fifth, as you got it correct, the phosphorylation of fructose 1,6 phosphate to fructose 1,6 bisphosphate is not correct. It is fructose 6 phosphate that is getting converted to fructose 1,6 bisphosphate. So that's why it's incorrect. And the last one is again correct. Phosphofructokinase 1 is a rate limiting step in glycolysis. So be mindful of the words that are being used. Irreversible, reversibles, or reversible, 1 phosphorus, 6 phosphate. So just be mindful of those words. Okay. Right. Now, so this is basically you have the cleavage of fructose 1,6 bisphosphate. So this step here, right? So what I skipped while discussing was this question. This question is important, or is this question is a good way to interpret or to look into that step. So basically what it says is there is 7 step here involved, and you have to arrange these following steps in the reaction mechanism of aldolase catalyzed cleavage of fructose 1,6 bisphosphate in the correct order, right? So if you think one happens first, write one happens first, then write four, then write five, then write whatever happens first, you write in that order. Read the read the answers, and then give me the correct answer. So look into the statements, and I will probably also keep this here. So this is the reaction that we are trying to see. 1,6 bisphosphate is broken down by this aldolase into two products, DHAP and glyceraldehyde 3-phosphate. So this is not a straightforward one single step reaction. This has, as you can see, at least 7 steps involved. So these 7 steps are enlisted here. If you are not able to do it, no problem. We will still wait for 1 to 2 minutes for you to read the statements first and try to think what's exactly happening, and then we will look into the answers. So the first one says CC bond cleavage leads to the release of first product and the formation of enamine intermediate. The first statement itself gives you an idea that it is already talking about release of one other product, right? Second step talks about formation of protonated Skiff's base by isomerization. Third talks about protonated Skiff base by rearrangement, right? Fourth talks about proton exchange with solution restores enzyme. Okay, so there is proton exchange and enzyme is restored. Fifth talks about binding of the substrate to aldolase and opening of the furanose ring. Sixth says Skiff base is hydrolyzed and the second product is used. The wording in few of the statements are very, very definitive. And then the 7 1 says active site lysine attacks substrate carbonyl, leading to the formation of a tetrahedral intermediate. If you just follow the words, you should be able to at least guess most of the uh, order or most of the series of the options. One more minute, and then we will look into the answers. In the meanwhile, you can look into the options and try to think what it would be, or at least try to guess the first step. That should be 573. Okay. Okay. So let me look into the answer. 573, you got it right. The next one was 1264. Let's look into this. So this is what's happening. This is your fructose 616 bisphosphate. So the first thing that is happening is your aldolase. So your substrate is binding to aldolase and opening. So the furanose ring of this compound or this molecule is opening. So your aldolase binds to fructose 1,6 bisphosphate and the structure opens up. That was cue of understanding that this is what's going to happen. Next, as again, you got it right. The active site of lysine of the enzyme attacks the substrate carbonyl, and this leads to the formation of a tetrahedral intermediates. Something similar to this we already saw in the previous slide when we were looking into the enediol intermediate, right? Third one is formation of a protonated Skips base by rearrangement. So this is that. Now what happens, as I have told you in the very first time that I read, that fourth state or the fourth in order sentence is CC bond cleavage leads to the release of the first product. So your first product is released here after the formation of protonated Skips base. So what is the first product? Glyceraldehyde 3-phosphate. Now whatever is remaining is what is forming a enamine intermediate. For that, what happens is you have a formation of protonated Skips base by isomerization, which is what happens here. This Skips base is hydrolyzed and a second product is released. And lastly, because you need your enzyme back, your proton exchange happens and your enzyme is back. So enzyme, remember, enzyme was bound to the substrate. You need the enzyme back. So your proton exchange happens and the enzyme is back in the solution, right? So up till at least the first four were already in the, if you read it, you would be able to, or I can see you've already tried to answer. So that was great. So you got it till 3. It was 2, 1. It was 1, 2. You have written 2, 1, but that's okay. But that's fine. So it just lets you know that you are thinking in the correct direction. And that was great. All right. So let's move on. We have already covered the first phase of glycolysis and we have looked into the several steps that are involved. Let's move on and let's look into the second phase, which is payoff phase. Now, what is payoff phase? You have prepared your glucose to form glyceraldehyde 3-phosphate, which is finally giving you the ATP. Now, how to go, how to reach till ATP? You've already used glyceraldehyde 3-phosphate. You have made it. Now, how to go back to it, pyruvate? Now, why is this called payoff? This is called payoff because the ATP that you used, let me go back to maybe one of those slides. So in the preparatory phase, you're using 2 ATP. In this phase, you are going towards producing those ATP. In the meanwhile, also returning the ATP that you use. That's different case scenario. But you are now going to pay off. You're now going to give. You're now going to produce ATP. So that's the payoff phase. So what is exactly or briefly happening here is, in brief terms, there is oxidative conversion of glyceraldehyde 3-phosphate to pyruvate, and then the coupled formation of ATP and NADH. So ATP and NADH. So payoff because you have used ATP in the first phase and you are now returning it back. Okay, that's why it's payoff. And it's, if you remember it that way, it's simpler for you to remember. Okay, now in what happens, which is the first step in payoff phase. What is the starting material? starting material is glyceraldehyde 3-phosphate. There are two molecules of glyceraldehyde 3-phosphate. We look into one. Whatever is happening for one molecule is also happening for the other molecule. Okay. So the first step here is oxidation of the glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. Basically, you are adding another phosphate to the molecule. So here the enzyme is glyceraldehyde 3-phosphate dehydrogenase. Okay. And because you are using a dehydrogenase, you are using the NAD. You are basically reducing the NAD plus, right? So the dehydrogenase is basically taking away hydrogen from glyceraldehyde 3-phosphate and giving it to NAD plus. So that's the first step. So what you also need to remember now, uh, you also, like I said, you need to remember how many molecules are there. You also need to remember how many ATPs are being used and how many NADH are being used. Today I think I have one equation that gives you the NADH and ATP used in glycolysis. But you should remember the formula of NADH and ATP in citric acid cycle, in glycolysis, and all the pathways that you look into, especially glycolysis and citric acid cycle. Okay, well, so the first step in the payoff phase uses NADH plus to reduce it to form NADH. In the meanwhile, your glyceraldehyde 3-phosphate is getting oxidized to form 1,3-bisphosphoglycerate. Right? The two that you see here is basically two. So there are two glyceraldehyde 3-phosphate. So everything is happening twice. Okay. Now, what is after that? What happens to the 1,3-bisphosphoglycerate? The 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate, right? And how is that happening? That is happening by the enzyme phosphoglycerate kinase, which transfers a phosphoryl group from the carboxyl group of the 1,3-bisphosphoglycerate to ADP. So this is the first step where ATP is generated. Okay. Now, what is this step called? This step is called ATP forming reaction or substrate level phosphorylation. Here, you are phosphorylating ADP by taking the phosphate from this molecule, bisphosphoglycerate, and adding that phosphate to ADP, which is making it ATP. So this step you need to remember. There are two substrate level phosphorylation. The first step is here, bisphosphoglycerate forming 3-phosphoglycerate. Okay, that's happening here. What happens after this? After this, your 3-phosphoglycerase is becoming a 2-phosphoglycerate by phosphoglycerase mutase. Okay, and again, this is reversible. The phosphoryl group is shifting between the C2 and the C3 of the glycerate compound. So this is glycerate. The backbone is glycerate. You have phosphate here, which now moves to 2. It was in the third carbon, it is now moving to the second carbon. And that's happening because of phosphoglycerate mutase. This is this enzyme here. This is again reversible. The ninth one. The ninth one is where in the 2-phosphoglycerate is converted to PEP, a very important compound in terms of for glycolysis, phosphoenol pyruvate, PEP. Okay, what is happening here is you have the enolase enzyme. This promotes the reversible removal of water molecule from glycerate, and that gives you phosphoenol pyruvate. Okay, because this is where you have the pyruvate already coming into structure. You just need to now remove the PEP or phosphoenol group and you will get the pyruvate, which is what you want to get. So phosphoenol pyruvate, right? The last step is where you have the phosphoenol pyruvate getting converted to pyruvate, right? This step again has a phospho group removal. So where does the removed phosphorus group go? It goes to ADP, which forms ATP. So one here and one here. This is the second substrate level phosphorylation. And which enzyme here? Pyruvate kinase. So what it does, it transfers phosphoryl group from the PEP to ADP and which forms the pyruvate. Now this pyruvate is in enol form, it tautomerizes and it forms the keto form, which is a stable form. This is irreversible and this is where you have, that's why you have the glycolysis pathway ending at pyruvate, which then enters other pathways to give you ATP. Okay, so the glycolysis begins with one glucose molecule and ends with two molecules of pyruvate. One glucose molecule gives you two pyruvate molecules. There are three irreversible steps. There are two substrate level phosphorylation. These things you need to remember. We'll see the equation. So let's see. This is what the glimpse is. So you have glucose, you have 2 ATP, which is being used. You have 2 NAD, 2 remember, because one glyceraldehyde 3 is using 1 NAD. So 2 NAD plus 2 ADP, and of course 2 PI, 2 inorganic phosphates. What are the result? What are the products? You have 2 pyruvates, 2 ADP here, 2 NADH, and 2 H plus, 4 ATP, and water molecule, which was here, which was removed from here. Now you have ATP here also, you have ATP here also. So just remove them cancel them. So 2 ATP removed. So final equation becomes, for the glycolysis, glucose plus 2 NAD plus 2 ADP plus 2 inorganic phosphate, giving 2 pyruvate, 2 NAD plus, H plus, ATP, and 2 water molecules. This final equation is what you need to remember. Okay. Whenever you're reading glycolysis, this is how you should remember the final equation, the final like, which gives you the stoichiometry of reactant and products, and the specific steps, which is where you need to remember what exactly happened. Okay. Now, so our glucose is now converted to pyruvate, and the pyruvate is supposed to give me ATP, but that can only happen under aerobic condition. What else is the fate of glucose, which is entering as pyruvate? So your pyruvate can convert to lactate, which is basically lactic acid fermentation. This is via the enzyme lactate dehydrogenase, or it can be converted to ethanol by fermentation. Fermentation happens under anaerobic condition. When there is no oxygen, you have either lactic acid formation or you have either ethanol formation. This is what people use. This is what is used in breweries, right? Ethanol, or you want to generate some lactic acid. This is what you are keeping in mind. So your pyruvate can be converted to lactate or ethanol via intermediate acetaldehyde under anaerobic conditions, right? If it is lactic acid formation, it is via lactate dehydrogenase, or if it is ethanol formation, there are two enzymes involved, which is one is pyruvate decarboxylase, and one of them is alcohol dehydrogenase. So this, you also need to remember, once your glucose has converted to pyruvate, what is the fate of the pyruvate? Under aerobic condition is what we are referring to as formation of ATP, but if there is no aerobic condition, these are the fates of pyruvate. So this is what I was referring to. So you have glucose, one glucose giving two pyruvate molecules. These two pyruvate molecules can either form under anaerobic condition or hypoxia. Hypoxia means less or deficit of oxygen. It can form ethanol in yeast, or it can form lactate, two lactate molecules. So where do you see lactate molecules? You basically would have heard about cramps in muscles. So the cramps in muscles is because of this lactic acid formation. And this is what is written here, that it vigorously contracting muscle or erythrocyte when there is anaerobic condition or when there is deficit of oxygen, you have lactic acid formation, and this gives cramps. Okay. But when you have aerobic condition, this is further converted to acetyl coenzyme A, and that gives rise to carbon dioxide and water, and in simultaneously it is also leading to the production of ATP. Okay, this is the flowchart that you should remember. Okay, we have discussed enough about glycolysis second pathway also. So let's quickly do these kind of questions that makes you get a better grasp on it. Right. So uh, so the step where glyceraldehyde 3-phosphate is converted to 1,3-bis phosphoglycerate, what all steps are actually happening in that reaction? So let me show you that reaction once again. Yeah, this is the reaction that we are talking about. Glyceraldehyde 3 to 1 3 bis phosphoglycerate. So what all is happening in this step? You can look into the name of the reactant and the product. That will give you the idea. Yes, you have one A, D, and F as the answer for happening, which is exactly what is the answer. A, D, and F. So let's see into this answer more detail. The first one says the addition of phosphoryl group, which is there in the name itself. You have 3-phosphate, which is now becoming 1,3-base phosphoglycerate. So there are 2 phosphate now. That's correct. Now, the second one says that there is a covalent interaction between sulfhydryl hydrogen and imidazole nitrogen at enzyme's active site, which is incorrect because this reaction, let me go back to the reaction. Yeah, here. So the reaction which is catalyzed by glyceraldehyde 3 phosphate dehydrogenase, it forms a thiohemiacetal intermediate, and this has a cysteine residue in its active site, right? So there is that cysteine residue is what is acting in actively part. The sulfhydryl hydrogen and imidazole nitrogen is not what actively takes part in covalent interactions. Okay, it is basically a formation of thiohemiacetal, and that is exactly what is in the fourth option. The third option says an aldehyde is converted to a free carboxylic group. That's not correct. The aldehyde group is oxidized, but it is not converted to a free carboxyl group. It forms an acyl phosphate. That is what is making it add have it when it gains a phosphate, it becomes acyl phosphate. Fourth, as we see, it is correct. It forms a thioacetal intermediate, which is oxidized by NAD plus. Right now, E says there are 2 molecules of NAD plus oxidized per molecule of glyceraldehyde 3 phosphate. Let me again go back here. One glyceraldehyde 3 phosphate, one NAD plus is reduced. There is no 2 molecules here involved. So this is wrong because when it says 2 molecules per glyceraldehyde, that's incorrect. One molecule is one NAD plus. 2 glyceraldehydes have two molecules of NAD plus. So that's correct. And lastly, there is a net gain of 2 electrons per molecule of glucose in converted in the form of NAD plus, which is correct because you have 2 molecules of NAD plus being reduced. Okay. Next. So which of the following statements are incorrect about the part of glycolytic pathway that leads up to the formation of 1,3-bis phosphoglycerate? So leads up to the formation means basically all the steps till 1,3-bis phosphoglycerate. So of course your preparatory steps, first few steps, and then the steps of payoff phase. So remember those steps and then try to answer this question. So the step of conversion till 1,3-base phosphoglycerate, there are 7 steps there. So you need to remember how many ATPs were used, how many NADH or NAD plus were used and formed. That's what the question is all about. B only correct. Okay. A, B, D. All right. So let me give the answer. Okay. So B is correct, definitely. D is also correct. And A, C, D is incorrect. Yeah. Let me tell you why. Okay. Do I have that overall? Okay. I will come back to this later. Maybe I'll go here. All right. The first one talks about for one molecule of glucose, net 2 molecules of ATP are formed. Let me go back here. This step is here, right? We have discussed in the previous thing. So what we are looking for is till this step here, right? And the first statement was about for one molecule of glucose, net 2 molecules of ATP are formed. Up till this step, no ATP is formed. ATP is being used, but no ATP is formed. So that's why that statement is incorrect. Coming back to the second statement, for one molecule of glucose, net 2 molecules of NADH are formed. Again, if we go back to this, till this step, NADH is formed. Definitely is, right? So it has used up 2 NAD plus, reduced it to form NADH. That's correct. Third one says one substrate level phosphorylation reaction occurs, and that is again correct. Sorry, that is again incorrect, because substrate level phosphorylation is the next step after bisphosphoglycerate, because your 1,3-bis phosphoglycerate gets converted to 3-phosphoglycerate, and in the meanwhile ADP is converted to ATP. So that's the first substrate level phosphorylation that occurs after the formation of bisphosphoglycerate, not till bisphosphoglycerate. Okay. Next, a total of 2 protons are consumed per NADH molecule form. That's correct, because you have 2 molecules being used. And the last one says that 2 molecules of NAD plus is oxidized. If your this statement is correct, then this statement is wrong, because NAD plus is reduced to form NADH. So that's why D and B both are correct. Okay. Okay. The last one. Now, from we have all done both the phases. Now, out of these 10 enzymes which are not part of payoff phase of glycolysis, which is basically the second phase. Try to remember all the different steps and then remember which where do you use them. C and E. C and E is what being used in the payoff phase. Oh, do not use in the phase of, okay. C, E, F and G. Okay. Okay. So yeah, C, E, F and G, right? Okay. Now let me go back here and then let me look into the answers. So this is what we did, right? So I will show you the answer to that question, the previous question, as in when we look into the enzymes here. So this is a snapshot, or this is the overview of what we discussed so far. We've discussed preparatory phase, 5 steps here. We have discussed payoff phase, 5 steps here. Okay. This Glucose here is converted to glucose 6 phosphate, converted to fructose 6 phosphate, 1,6 bisphosphate, which gives you two molecules, glyceraldehyde 3 phosphate and DHAP. This DHAP is isomerizing and forming two molecules and isomerizing and forming glyceraldehyde 3 phosphate, which finally gives you two glyceraldehyde 3 phosphate. Now, these two glyceraldehyde 3 phosphate is what is being used in the payoff phase. So, in payoff phase, you begin by uh, formation of 1,3 bis phosphoglycerate that gets converted to 3 phosphoglycerate, then 2 phosphoglycerate, PEP, and pyruvate. Right now, the enzymes that were asked these questions, the enzyme that was asked, enolase. Where is enolase? Enolase is here. So it is used in the payoff phase. Then we had pyruvate kinase. Where is pyruvate kinase? It is being used in the payoff phase. Then we have aldolase. Where is aldolase? Here in the preparatory phase. That's why it's incorrect. Phosphoglycerate mutase is here. So it is used in the payoff phase. Phosphofructokinase, we had seen into this. This was used in the preparatory phase. Then we had glyceraldehyde 3 phosphate hydrolase. Glyceraldehyde 3 phosphate hydrolase. You don't see that. Glyceraldehyde 2 phosphodehydrogenase. Glyceraldehyde 3 phosphodehydrogenase. So the name of the enzyme also you should take care of. This is incorrect and this one has a wrong number attached to it. If it was glyceraldehyde 3 phosphate dehydrogenase, that would be part of payoff scale. It's not what is there. And lastly, phosphoglycerate kinase, which is here. So it is also part of payoff phase. So this is what you need to CEGH. Yeah, CEGH. H is phosphoglutarate kinase, which is part of the seventh reaction, which is here. The first substrate level phosphorylation is because of phosphoglycerate kinase, right? Okay. Now, since we are closing this topic, what you need to remember in glycolysis is, like I have been mentioning, which ones of these steps are irreversible, which one of these which one of these steps are irreversible, which are these priming reaction, which are the substrate level phosphorylation, and which is the what is the enzyme that is used in all of these steps. Okay, and most importantly, how many ATPs and how many NADH are used and produced. That equation that we did, this equation, this you should remember. Okay, that the more you read, the more grasp on glycolysis should come to you. Okay, now let's move to the next topic, which is gluconeogenesis, which is very in very uh, easy terms. If glycolysis is breakdown of glucose, gluconeogenesis means formation of glucose, new glucose. So neo means new glucose genesis. Genesis means to create. So you're creating new glucose. So you're basically uh, like coming back from pyruvate to glucose, right? So ideally it should be exactly opposite of glycolysis, but that's not the case. And why is that not the case? Because in glycolysis, you have three irreversible reactions. And we know irreversible reaction means it can only go in one direction. It cannot come back in the other direction. So at those steps, the three reversible reaction, the gluconeogenesis pathways take a reroute or an alternate route. And that's why gluconeogenesis and glycolysis are not exactly opposite of each other. Other steps, they are reversible, they can go back in the other form, but the three steps make it not identical, right? And we can see that here. So to come to glucose, I should start from here. So remember, PEP to pyruvate was an irreversible reaction. So pyruvate forming PEP is via the intermediate oxaloacetate. There are two enzymes that are involved here, pyruvate carboxylase and PEP carboxykinase. So it is getting converted to pyruvate by one enzyme, but for it to convert back to phosphoenol pyruvate, it has to take two enzymes and one intermediate. Okay. Then when you have PEP or phosphoenol pyruvate, it gets converted by the same enzyme enolase to 2-phosphoglycerate, via the same enzyme phosphoglycerate mutase to 3-phosphoglycerate, via the same enzyme phosphoglycerate kinase to 1,3-bisphosphoglycerate, and you have glyceraldehyde phosphate dehydrogenase forming glyceraldehyde 3 phosphate, right? Again, you have triose phosphate isomerase to break it up into two forms, dihydroxyacetone. So it get isomerized and forms these two components, which again via aldolase form fructose 1,6 bisphosphate. Now, fructose 1,6 bisphosphate formed by a PFK1 from fructose 6 phosphate. But remember, this was an irreversible reaction in glycolysis. So to go back to fructose 6 phosphate, the enzyme that is used here is fructose 1,6 bisphosphatase, not phosphofructokinase. So this step here is again different. Then you have isomerase that makes it 6 fructose to glucose 6 phosphate, and then instead of hexokinase, you are using glucose 6 phosphatase. So what you need to remember, and how can you make it easier for you to remember? You need to remember the three enzymes that are unique in gluconeogenesis, or four enzymes for that matter. What are those enzymes? Glucose 6 phosphatase, first step, fructose 6 1 6 or last step, fructose 1,6 bisphosphatase in the third last step, and the first step of gluconeogenesis, pep carboxyl kinase and pyruvate carboxylase. This you need to remember. You remember maybe glycolysis, full 10 steps, all the products and substrate, and then you just need to remember for gluconeogenesis what are the uh, enzymes which are different. That makes it easier for you. That's just a cue. All right. So we so when I was talking about regulation and enzyme, I had talked about a lot of regulation that we will do in the coming slide, I believe. So before that, we'll finish this also. So now that we have looked into gluconeogenesis and glycolysis together, let's try to answer these questions. So identify the incorrect statements now from these given options. You first try to attempt and then I will give you the answers. Okay. Okay, I'll wait. A, B, C, D. A, B, C, D are incorrect. Okay, incorrect is the question, right? Yeah, okay. EFG is only correct. Oh, wait. A, B, C, D, F, G, H, E, F, G. Okay, okay. Yeah, I think in my answers also I've got one wrong thing, but oh, sorry. Okay. So here it is A, B, C, F, G. Sorry, G, H. What did we write? We wrote F, G, H. E, F, G is correct. E, F, G is correct, right? E. Sorry. and we wrote A, B, C, D, F, G, H, A, B, C, D, F, G, H. Okay. So a mistake here, of course, is the D. As I can see, I didn't put it for some reason. I don't know why. I'll come to that. Right. So we are looking at the incorrect statements. So the first one is incorrect. Conversion of glucose to pyruvate gives 3 ATP molecules. Our equation showed that it's not 3 ATP, it's 2 ATP molecules that's generated. Right. Fermentation is a process of generating energy from proteins without the use of oxygen. No, you are using pyruvate, not protein. Pyruvate decarboxylase is present in the human muscle cells. Let me go back to this slide here. Where is pyruvate decarboxylase? It is in the formation of ethanol. So you are not forming ethanol in human cells. In human cells, under anaerobic condition, lactate dehydrogenase is acting, which forms lactate fermentation, lactic acid, and that, as I have mentioned, it in athletes it gives cramps, right? Okay. Then net gain of ATP from the conversion of PEP to pyruvate per molecule of pyruvate is 2. That's incorrect because here. So per molecule of pyruvate is 1 ATP. So this should be 1 or it is incorrect. So this answer is also incorrect. Next, thymine pyrophosphate carries active aldehyde groups and the E and F both are correct. Okay. And lastly, someone said G is correct. Well, G says pellagra. Pellagra is caused by vitamin B3 and thiamine is vitamin B1. So that's not correct. Lastly, lowering of glucose level in blood is called high. Lowering is hypo. So hypoglycemia. If it's hyper, it's more. Sorry, not D. Not D. Not D. D is correct. Okay. F is carbocation. That's exactly what's written, right? Oh, wait. What did it say? The two in the thiazide dissociated protein forms a carbocation which readily reacts with. Okay. Yes. Right. I'm sorry. So this one is the only correct answer. This F is should be carbo and ions. Sorry, I'm extremely sorry. So this and this are also incorrect. Thanks for pointing that out. So once again, the incorrect answers are A, B, C, D, F because it is carbo and ion, and G and H. Right? Thank you for pointing that out. Okay. Yeah. Next, this one. So, which of the following pathways can give rise to glucose? Read the statement, read which say pathways they are referring to, and yeah, which one is correct. A, B, C, D, E, and C, D. Okay. So A, B, C, D, E, and C, D. Okay. Let me give the answer. C, D, you most of you got it right. B also, I think one of you answered correct. A is incorrect, and E also is incorrect. Let me show you how. Okay, so it says lactate to pyruvate, which then passes through the TCA to give glucose 6 phosphate. Well, so you have lactate to pyruvate, which is via lactate dehydrogenase. But if that's happening, it's going to give rise to gluconeogenesis. That's correct. But this, if it goes to TCA, if pyruvate enters TCA by any chance, it doesn't contribute to glucose, because in TCA, as we know, TCA gives rise to carbon dioxide, and TCA is what we saw here, right? Let me go back to this thing here. This is your TCA cycle. And if you have pyruvate entering this step here, it gives rise to carbon dioxide and water, and simultaneously ATP generation. But in TCS, if TCA cycle is what the pyruvate is entering, then it will not form glucose. And that is why that is not the correct answer. Um, sorry, I'm sorry. I'm doing so much to and fro that it's missing my flow is missing. Right. B also, I think someone got it right. So carbon dioxide fixation by plants gives rise to 3-phosphoglycerate, which can be converted to glucose 6-phosphate, and we have seen this already. And this cycle is called Kelvin cycle. Okay. Third one is hydrolysis of TAG to glycerol, and glycerol then to glucose 6-phosphate, which is possible. D, glucogenic amino acid through the TCA, which can again give rise to glucose 6-phosphate. Uh, this is via intermediates like oxaloacetate that we will do in the coming slides quickly. And last, someone wrote E also, that E is incorrect. And that's because free acid or fatty acids, they cannot be converted to glucose because acetyl coenzyme A. So if your fatty acid gets oxidized, it forms acetyl coenzyme A, and acetyl coenzyme A again enters TCA. TCA gives carbon dioxide and water and in your ATP. So free acid gives you acetyl coenzyme A. That's why you cannot have glucose. And similarly, because in both of these you have TCA as the pathway that is being followed, so you don't have glucose after that. Well, so that brings us to an end of glycolysis, gluconeogenesis. And what you need to remember for path regulation, which I thought it's part of this, but it's not. Well, regulation for any of these pathways in very broader sense is mediated by the concentration of the reactants and the products. If your products is too high, then then forward reaction is inhibited because the product is too high. But if your product is less, then you are in need of those products, so your reaction will take place in the forward direction. For example, let me quickly just give you one brief example. Where's the enzyme? Here, phosphofructokinase. This here, right? This fructose 6-phosphate gets converted to fructose 1,6-bisphosphate. And if you remember, this is one of the committed steps of glycolysis. Now, this is highly regulated step. If you have more of fructose 1,6-bisphosphate, then this step is inhibited. And this step inhibition means there is no more fructose 1,6-bisphosphate formation. If you have less of fructose 1,6-bisphosphate, there's a demand in the cell for more ATP, more energy. So this reaction will take place, right? So your reaction, they will do it in a citric acid cycle, I'm sure. Well, I will show you this same thing if it is not becoming clear. If there is need, if there is deficit of a certain component, for example, ATP or the product or any other thing that links to that product, then the reaction will take place. Otherwise, if they are in excess amount, the reaction is controlled, it's regulated, it is stopped. And that's how you have optimum concentration of all your metabolites in the cell. Otherwise, if there is no regulation, if there is no checkpoint, then these reactions will keep taking forward and there will be no regulation. There will be everything in excess, and that's not what cell desires. That's not how cell will function properly. So for cell to function properly, everything should be at optimum condition and at desirable concentration. And that is why all these enzymes, the major enzymes that are committed to glycolytic pathways are highly regulated. One of them is phosphofructokinase 1. Okay. All right. So this you should remember. Right. Let's move to citric acid cycle, which is what happens after you get your pyruvate from uh, the glucose. So here on the left, there is a 3 vertical bar. The first bar here till pyruvate is what we have studied. So from pyruvate, you have the formation of acetyl coenzyme A, which is by 3 complex enzyme, which is called, or an enzyme which has 3 enzyme complexes, pyruvate dehydrogenase complex, which uses NAD plus, TPP, lipoate, FAD to finally give NADH and acetyl coenzyme A. This is how the enzyme looks like. Now, this acetyl coenzyme A is not only formed by glucose pathway or glycolytic pathway. It can also be given by fatty acid. That is something that I just discussed. It can also come via amino acid. But the main thing is you'll need to have this acetyl coenzyme A that enters the citric acid cycle. Right? This acetyl coenzyme A is used in the formation of citrate from oxaloacetate. So while you have oxaloacetate, which is converting to citrate, this acetyl coenzyme A is used there, right? So it's basically getting oxidized. Now, it is used in this cycle, and while it is being, as we will see, the steps are taking place. You have this NADH2, FADH2 taking forming, and this is what will ultimately lead to the formation of ATP. So right now we have already done the first step. We will now look into the second step, right? This is how you connect all the 3 major pathways that we will be looking into. So you have acetyl, first of all, your pyruvate formation. From pyruvate, you have acetyl coenzyme A, and from acetyl coenzyme A, we will then move on to go to ATP generation. So pyruvate is converted to acetyl coenzyme A by pyruvate dehydrogen complex, which is basically 3 enzyme or 3 complexes together. The first complex here that you see is pyruvate dehydrogenase E1. Second one is dihydrolipoyl transacetylase, and third is second is dihydrolipoyl transacetylase, and third is dihydrolipoyl dehydrogenase. Okay, very complicated names it seems like, but when you understand the reaction taking place, it becomes easier. So the starting material is pyruvate. What is the first enzyme here? Pyruvate dehydrogenase. It dehydrogenates the pyruvate. And what happens here is your TPP is getting ethylated. Okay. And this is then connected to the second enzyme, which has acyl lipolysin, which is basically getting oxidized, which is then again taking part, going to lysine. And while this all step is taking place, there is this formation of acetyl coenzyme A. Simultaneously, your NADH is also being synthesized. So the end product of this first step from pyruvate to acetyl coenzyme A is also generation of NADH. Right? Now your acetyl coenzyme A enters the citric acid cycle. What it does. So like I said, this acetyl coenzyme A is used in the step where oxaloacetate is converted to citrate via citrate synthesis. You need this acetyl coenzyme A, otherwise this reaction will not take place. And if this reaction doesn't take place, we won't have this NADH and FADH, which are the currency that will be used for ATP generation. So you have acetyl coenzyme A. This is used while the oxaloacetate is converted to citrate. This is a condensation reaction. Then, so you can see here. So the first step is where your oxaloacetate combines with acetyl coenzyme A, gives you a citrate. The enzyme in use here is citrate synthase. Second is when you have the citrate getting converted to isocitrate via an intermediary form of cis-econitrate. Enzyme used here is econitase, and the reaction is first dehydration and then hydration. So first you have a removal of water molecule, and then you have a water molecule which is being added, right? So this is how it takes place. Third is when you have your isocitrate getting oxidized to form alpha ketoglutarate and carbon dioxide, right? This enzyme is isocitrate dehydrogenase, and this is oxidative because it is using oxygen, and decarboxylation because you are removing carbon dioxide. What else you are also generating here is NADH. First step where NADH is generated. Okay. What is next? Next is your alpha ketoglutarate is further oxidized to succinyl coenzyme A. During this time, you are releasing oxygen. This is again an oxidative decarboxylation step, and this is by alpha ketoglutarate dehydrogenase complex. Complex means there are several enzymes that have come together to accomplish the reaction. This step also leads to the generation of NADH. Okay. The next one is succinyl coenzyme A getting converted to succinate. So you basically are removing the coenzyme A, and that is by synthetase. Succinyl coenzyme A synthetase. Here, you can either use GTP or ATP. So again, see this thing, again it's here. When you have a GTP or ATP formation, it is called substrate level phosphorylation. So there is also a substrate level phosphorylation in citric acid cycle, right? After that, you have this succinate that forms the fumarate via dehydrogenation. Now, when this dehydrogenation is taking place, you have FADH2 forming. Okay. The next step is this fumarate converted to malate. How? By hydration. And this is by fumarase. The next step is malate converted to oxaloacetate by again dehydrogenation. What is important? It also gives rise to NADH. So there are 3 NADH and 1 FADH2 per acetyl coenzyme A. How many pyruvate comes? 2 pyruvate. 1 pyruvate, 1 acetyl coenzyme A, 1 acetyl coenzyme A, 3 NADH and 1 FADH. These numbers that I'm telling, this many NADH and this many FADH is very important. This is what will come in your come handy to you when you are at the end, when we do ETC, when we will calculate the number of ATP generated. These currencies will come into play. So while you are studying, keep in mind these. While you are studying, keep in mind these numbers that you are encountering. So you have 1 acetyl coenzyme A from 1 pyruvate that enters citric acid cycle. It gives you 3 molecules of NADH from 3 successive steps and 1 FADH2 molecule. Okay. Right. And now lastly, you have the oxaloacetate formation. Again, you have oxaloacetate going back to citrate. So this is a cyclic step, right? But why is it important? How does it connect glycolysis with, how does it connect with glycolysis? It connects via acetyl coenzyme A that is formed by, or that is a product when pyruvate is converted to acetyl coenzyme A. Okay. All right. Now, this is what I was talking about when I was talking about the regulation of glycolysis. Look at here. So this is pyruvate. This got converted to acetyl coenzyme A. That acetyl coenzyme A entered the citric acid cycle. And then all this reaction took place. Now, there are some key checkpoints where the citric acid cycles are regulated. What does it mean? It means that at this steps, the decision to go ahead or not is taken based on the concentration of the products in the cell or the intracellular concentration of these components. Now see, if ATP, acetyl coenzyme A, NADH, fatty acids are present, then this step is not required. Why? Because ATP is high in number. You already have acetyl coenzyme A. You don't need any more acetyl coenzyme A. You already have ATP. So why do you need any more ATP? You already have NADH. You already have fatty acids. So you have everything in optimum concentration. You don't need this conversion, because in this conversion takes place, it's basically leading to the formation of all these components. But these components are already present. So if these components are higher in number, you don't have these reaction taking place. But now see, if you have the subset of these components, or if you have the monomeric units of these components, for example, AMP, which is adenosine monophosphate, or just the coenzyme A, or just NAD plus, or just Ca2 plus. If you have more of these in the cell will start asking for ATP. The cell will be deficient of acetyl coenzyme A, no energy currency. That's when your these steps will be regulated towards the forward direction. Because you have more of AMP, you now need ATP generation, so your acetyl coenzyme A will take place. So on the same grounds, your this step where acetyl coenzyme A is incorporated into the citric acid cycle, that step is also regulated by the substrates in the cycle itself. Succinyl coenzyme A citrate, where were they? They were here. This is citrate. This is succinyl coenzyme A. These two concentration determine if acetyl coenzyme A needs to be incorporated. Why? Because if citrate concentration is higher, you don't need this conversion of oxaloacetate to citrate. And that determines that happens because of incorporation of acetyl coenzyme A. But if you already have citrate very high in number, this reaction is inhibited. So you don't need your acetyl coenzyme A entering into your citric acid cycle. Similarly, citrate, similarly, succinyl coenzyme A, similarly, NADH, which is again a product of this cycle. So the products, the major products of the cycle will determine if the cycle moves forward in its direction or if it is halted. At every major step, for example, here, isocitrate, it is regulated by ATP. Alpha ketoglutarate is regulated by the immediate next product, which is succinyl coenzyme A. So when you go back here, succinyl coenzyme A not only controls or not only regulates the reaction, which is what it is forming from, which is its immediate substrate, but it also regulates the starting point of the reaction, which is the did you am I audible now? Was I unmute for a long time? Were you able to hear on the last slides? Which slides did you not hear? Okay, let me show you the slides and let me tell me which slides were you not able to hear. You could have messaged me. I would have seen the messages. Did you listen to me on these slides? The slide? Okay, this one. All the different steps of citric acid cycle. All the steps were covered. Were you able to listen to all the steps? Okay. So you did not listen to the regulation of citric cycle at all? Okay. No issues. We have time. We'll cover it up soon. Okay. No issues. So sorry about that. I don't know how I went to mute. All right. Okay. Well, uh, I will just quickly cover it once again. I don't know how long I have been on mute. So while what I was talking about was that in your citric acid cycle, you have your acetyl coenzyme A, which is forming from pyruvate, which is in the previous step that we have just done. Right? This acetyl coenzyme A gets incorporated into the citric acid cycle and it undergoes these different steps. So formation of citrate, then from citrate you have the isocitrate formation, then you have ketoglutarate, then you have succinyl coenzyme A, succinate, fumarate, malate, and finally oxaloacetate. Okay. Now, there are some steps here, for example, this, and this, which leads to the formation of NADH. And you have one step here, which is the succinate to fumarate step, which gives you FADH2. Okay. So, one pyruvate, when it converts to one acetyl coenzyme A, and that gets incorporated into the citric acid cycle, you have three NADH molecule and one FADH2 molecule. I don't know if you heard or not. I said that you should remember the numbers of these FADH and NADH because this will come handy when later stage we calculate the number of ATP generated per glucose molecule. So the number of ATP, the number of NADH generated, number of FADH generated is important when you are. So when you're looking or when you're studying glycolysis, keep these numbers in mind. When you're studying citric acid cycle, keep these numbers in mind. The currencies NADH, FADH, and ATP. Here also, you have a step where there is production of GTP or ATP, Again, we have already done in glycolysis, if there is a production of ATP or GTP, that is called substrate level phosphorylation. But that thing also happens here, right? So this is what I was telling. Now, the slide that I was trying to explain here is regulation of citric acid cycle. Now, when I was talking about regulation of glycolysis, I told you that the concentration, intracellular concentration of metabolites regulate if the step has to take place or not. And this will be apparent from this step here. So the conversion of pyruvate to acetyl coenzyme A is regulated negatively by ATP and positively by AMP. Why? Because ATP, acetyl coenzyme A, NADH, fatty acids being high in concentration or being present in the cell means that you don't need any more ATP. You don't need acetyl coenzyme A. So why do you need to do this step which leads to production of more acetyl coenzyme A? If you have acetyl coenzyme A, this step is not required. So cell will stop the reaction at that phase. So if you have acetyl coenzyme A, there will not be this step. If you already have ATP, then why should you bother to convert pyruvate to acetyl coenzyme A, which will enter this step, which will then further also generate ATP? You already have ATP, so you don't need this step. Similarly, you have NADH, which is the product here. Fatty acids, you already have all these components in concentration, which is optimum for the cell. So this step is not required anymore. So this step is regulated and this will not go in the forward direction. But if you have the monomeric unit of these very compounds, so AMP is basically adenosine monophosphate or coenzyme A or just NAD plus, if these components are present in the cell, it means the cell needs more of ATP, the cell needs NADH. How will these components come? If your acetyl coenzyme A enters citric acid cycle. So cell will decide, okay, let's convert pyruvate to acetyl coenzyme A. So the substrate getting converted to product is determined by the intracellular metabolite condition. And there is not only one checkpoint for any cycle. Here in citric acid cycle, you can see that there are at least 3 checkpoints. So let's say you have acetyl coenzyme A, but acetyl coenzyme A might not get incorporated into citric acid cycle. How does it get incorporated? When your oxaloacetate is converted to citrate. This step is inhibited if you have NADH, which is the product here, if you have succinyl coenzyme A, or if you have citrate, which are all the products already of all the substrates of these cycle. If the substrate of the pathway are higher in number, then the pathway doesn't have to go forward. So succinyl coenzyme A and citrate concentration determines if your acetyl coenzyme is incorporated into the citric acid cycle or not. Similarly, they also regulate the step that leads to their formation. For example, succinyl coenzyme A regulates if there has to be the conversion of ketoglutarate to succinyl coenzyme A, or ketoglutarate, ATP determines if isocitrate should convert to alpha ketoglutarate. What basically the idea is that I have already mentioned, that there is a certain amount that is required by the cell. And if that amount is present, then the step which are leading to the formation of those products will not be required by the cell and those steps will be halted. That's exactly what I was trying to tell. So this is an example of regulation of citric acid cycle. This same principle is followed for regulation of glycolysis, okay, where you have certain checkpoints which are controlled, one of which I already told you, phosphofructokinase 1. That step is controlled, that step is regulated based on what is the concentration of metabolites in the cell, right? Okay, so well, that brings a lot of talking to an end, but we have questions that should give you overview of the topics that we have done today. Let's try to solve them. That shouldn't be very difficult, but if it gets that, we'll discuss and get to the solution. Right. The first one here is, which of the following statements are true about TCA? TCA or citric acid cycle are basically the same thing. So you read the statements and let me know which one is correct. Is it difficult? You're still answering. No worries. I'll wait for, yeah, maybe. BCE. Okay. C is correct. E is correct. B is not correct. Okay, let me give you the answer. Okay. So we are looking at the correct statements for related to tricarboxylic acid cycle, right? So the first one says acetyl coenzyme A is reduced in the TCA cycle, which we know is incorrect because acetyl coenzyme A is oxidized, not reduced, right? And what it does, on the contrary, it donates electrons that are transferred to NAD and FAD to they are reduced to form NADH and FADH. So we have seen this already. Okay, the second one, glucose is the only source. That's not correct, right? That's the first thing that we did. Acetyl coenzyme comes from different other source also. The glucose is not the only source, right? Right. Next is oxidation will mean here the electron flow from acetyl coenzyme A to NAD plus and FAD. That's correct. That's exactly what it means. Because it getting oxidized means it's getting the electrons out and giving it to NADA plus and FAD for them to get reduced to NADH and FADH. That's correct. D says electrons are transferred from NADH and FADH to oxygen via oxidative phosphorylation. We did not do it. That's why it's okay. Well, this is correct. We will do this in the later classes where we look into electron transport chain and we will see how that reduces oxygen to give water. Okay. Next, E, electron transfer occurs in mitochondria, which is correct. So the ETC happens in mitochondria. And lastly, the electron gradient is used to make NADH, which is again, well, it is incorrect, not correct. I don't know why it didn't go. I don't know. So, well, this is incorrect because it's not electron gradient, which is used to make NAD plus. Okay. So the proton gradient is used to make, if we are talking about gradient, the proton gradient is used to generate ATP. NAD plus, as we will see when we do ETC, it is regenerated when NADH donates the electrons. So it's, if you're talking about gradient, it's about proton gradient when you're talking in terms about ATP generation. Okay. Well, next. So coenzyme A, which are the following are the components of coenzyme A? C. Okay, which is ribose 3-phosphate. Okay. Well, the answer is B, D, and H. Okay. So let me tell you the primary constituent or primary structure components of coenzyme A. I'm just trying to find the how to write. So if you look into coenzyme A and you look into their structure, you have pantothenic acid, which is basically vitamin B5. Then you have adenosine 3-phosphate. Okay. And the third component is beta mercaptoethylamine. So those are the major three components. Those are the structural components of coenzyme A. So out of given structure, you already have, given options, you already have pantothenic acid. You can have adenine because that is also part of this adenosine 3-phosphate. And then you have the 3-phosphoadenosine triphosphate, which is again part of these uh, structure, right? Okay. Next, this is very interesting question. So, which of these pairing is correct? This basically which enzyme pair for the reaction given is correct. Should not be very difficult. Should be a very easy question. And this kind of question definitely you will encounter somewhere or the other in some of the other questions exams. So you have to give me the correct statements. B, C, D, E, F, I. Okay. B, C, D, E, F, I. Fantastic. You got all of them right. So the first one, citrate to isocitrate, is via aconitis enzyme. Yeah. B, C, D, as we see already correct. G says three NADH molecules are reduced. NADH is not reduced. NADH is produced. So NAD plus is being reduced. And H says net three molecules are consumed per cycle, which is not the case. If you look into the cycle, there are several steps where water is consumed and water is also given off. So if you look into this cycle, you will see that it is less than 3 water molecules is being consumed. Okay. And lastly, again, you have all got it all right. Fantastic. That was great. Okay. Quick, I think there are 2 or 3 questions more and then we will be done. Again, next questions. Which of these following statements are correct? ACEF. Um, ACENF. Okay. Well, the correct answer is basically everything except for B. And that's because the B one says that succinate dehydrogenase is attached to the outer membrane of the mitochondria. When we will look into ETC, we will see that it is actually embedded in the inner mitochondrial membrane. So it's not on the outer membrane. While the others are correct, so delta G for succinate to fumarate is 0. If you look into the cycle, you will find this information incorporated there. Maybe I will go back to it and try to show you there. Yeah. Here, succinate to fumarate is 0. Delta G is 0. Then the next statement was from succinate to fumarate, the electron transfer is in the form of hydrogen. So let's look into this step here. Succinate to fumarate, you are removing dehydrogenation is there. So there is a removal of hydrogen. So that's electron transfer is in the form of hydrogen. Next is fumarase, which is this enzyme here. It is stereospecific and takes L-malate to form fumarate, or in the reverse direction. So it's stereospecific, and this is correct answer. E1, E talks about ATP being total number of ATP generated is 30 to 32. That I told you, 30 to 32 is what we will determine in the next class by the calculation how we got 30 molecules, 30 ATP molecules. And the last one is intermediate in the TCA serve as precursor. That we know. We have already seen that all of these, metabolite can serve as a precursor or serve as a substrate for different biosynthetic pathway. For example, your citrate here, where is it? Yeah, it is used in fatty acid synthesis. Your alpha ketoglutarate, you will see in amino acid synthesis. You will see succinyl coenzyme is something called heme synthesis, and oxaloacetate can also be part of gluconeogenesis. So you see different pathways have used these different metabolites from different other pathways. So all the pathways are somehow or the other connected and linked with each other, right? Yeah, well, you got all of them correct except for the second one. I mean, D1. Right. Right. And I think the last question for the day. So out of these option, which one is incorrect? B, C, D, E. Just a second. Let me navigate to my options. Okay, sorry. You say B, C, D, and E are also incorrect. Okay. Okay. Well, the first one, PEP carboxylase is a regulatory enzyme that we had established already. We had looked that into detail for that matter. Next is pep carboxy kinase is present in heart and skeletal muscle. That is actually correct. Did you say that was incorrect? That is correct. That is present in heart and skeletal muscle. Next is pep carboxylase is present in liver and kidney and is involved in gluconeogenesis. Well, that is incorrect. So the enzyme, as we know, is pep carboxykinase in gluconeogenesis. Pep carboxylase you find in plant, and they are involved in photosynthesis, C4C CAM photosynthesis. Okay. Malic enzyme is limited to prokaryotes. No, we find them in eukaryotes as well. Okay, that was correct. Just a second. B, C, D, E. Yeah. E also you said incorrect. Biotin acts as a carrier to transport CO2 from one active site. Well, that is correct because it is a coenzyme and it actually takes part in the transfer of CO2 uh, in carboxylation reactions. For example, the enzyme pyruvate carboxylase. So there you find biotin as coenzyme. And lastly, the overall end product regulate the enzyme in TCA cycle negatively. This we have established already. If the products are there, then it will stop the reaction. It negatively regulate means if they are present, the end products are present, they will stop the reaction because they are in high concentration or they are already present, so they will not. There is a no requirement for the reaction to take forward, right? So they negatively regulate the cycle that they are part of, right? So you got all of them correct except for one or two, but that's all right. That's not a big problem. But yeah, so that's it about today. I know it was a big class. It was a more of text and more of conversation. Well, these topics are like this, and it might get a bit tedious when you are reading, but that's okay. The more you read these topics, the more easier it becomes, or at least the more grasp you get over these topics, right? So next class also, I think we will cover ETC and we'll look into ATP generation, and then we'll move forward to other topics, right? Okay, so well, that brings us to the end of today's session. If you have any doubts, let me know, or else we'll meet each other in the next Tuesday at the same time. Thank you so much. Thank you for joining.